Thank you, Lee. So our last speaker is my fellow moderator, Natalia Vilor-Tejedor. She is a team leader at the Genetic Neuroepidemiology and Biostatistics team in Barcelona Beta Brain Research Center, and she'll give the final talk on multiphenotype analysis in brain imaging genetics of Alzheimer's disease. Thanks.
Thank you very much, Davia, for the introduction, and thanks you all for being here, and also thanks the organization of the OHBM for giving us the opportunity to present this symposium on this relevant topic for us, and I hope for all of you after this session. So in this presentation, what I'm going to try to provide is a summary of statistical strategies that we can, let's say, use in the context of imaging genetics, and I will try to focus also on multiphenotype and multivariate analysis. So as the previous speakers were introducing, brain measures are good targets for genetic analysis. In the context of Alzheimer's disease and also in other neurological and neurodegenerative diseases, we, let's say, have two main points for using brain imaging as our endophenotype of interest. The first one is mainly the heterogeneity of these diseases. So it's true that when we try to classify clinically cases and controls, there's misclassification in both groups. So this also makes difficult then the assessment of risk factors or genetic factors associated with these diseases. The other thing is that when we want to analyze or study the preclinical phases of the disease, it's true that we don't have this status, the disease status. So also these brain features that are altered before the symptoms appear are good targets for this analysis. And actually, when we go through the statistical analysis, the idea is to try to have in mind how to classify this data. Normally, when we work with the mathematical models, we are going to think on two main matrices. One is going to be our predictor matrix, that is going to be the genetic data. So in each row, we are going to have our individuals. In each column, we are going to have our genetic variants. And then we are going to have a response matrix that is going to be actually our brain features, and it's going to depend the units on the MRI or neuroimaging modality. So the idea is going to try to see if we find genetic variants like X matrix components associated with these Y matrix components. And to do that, we can follow different strategies. As I was mentioning and other speakers were mentioning, we can follow, let's say, a univariate analysis, which is going to be mainly focused on selected a target region, a target brain feature, and a target genetic variant. Here in this context, what we normally apply is Gaussian model, a linear regression model using Gaussian distribution, because normally these features are distributed in that way. It's true that there's other type of features that are not following this distribution, then we need to apply non-parametrical methods. But in this case, what we normally do in imaging genetics is to start by this. So basically what we are going to have is our effect and our p-value in most of the cases to see if these genetic variants, these SNPs that we were introducing before, are associated with this phenotype of interest. In the context of Alzheimer's disease, what we normally try to do is to focus on target regions associated with the disease. So mainly hippocampal volume is the target region in these kind of studies. And also, when we start to work in the context of Alzheimer's disease, you can see in the literature that we start focusing on genetic variants that are associated with Alzheimer's disease. So for instance, ApoE and ApoE4 status or parentship. So this is mainly what it has been done in imaging genetics. Also, as the other speakers were introducing, when we want to really discover novel markers, what we go and we follow is a genome-wide association study. So in this case, it's mainly the same that before, but reproducing these univariate models several times. At the end, we're going to perform a univariate model for each of the genetic variants that we have in our data set, which is a lot. And this is good for discovering new variants, but also has some problems, like, for instance, the multiple comparison correction that is going to probably affect the statistical power of our analysis. So as a starting point, it's a good strategy to go through this univariate and massive univariate analysis. But then we really need to focus on try to increase the statistical power of an analysis and also understand better the biological mechanisms. Because here in these models, the problem that we have also is that these genetic variants explain low variability of our phenotype, clinical or brain features. So we really need to understand better what is happening there. So in this sense, we can go through a multivariate analysis. And here I'm going to present two different strategies. So we can just perform a multivariate combination of data in the site of the genetics. We can go then through the multivariate analysis of both modalities. In this context, for instance, Luigi was introducing the PRS concept. What we are going to mainly do is to summarize all the genetic information that we have into one score, which is this PRS. And this PRS is going to summarize the genetic predisposition to a disease, can summarize also the genetic predisposition to a biological pathway in which these genetic variants are involved. And at the end, what we are going to do is to go again to the univariate model, but instead of performing these large or massive univariate models for each of the SNPs, we are going to perform just one model for each of the PRS or for our PRS. This is going to reduce a lot the computational efforts and also is going to increase the statistical power. But as I was saying, the idea here is to try to combine both types of information. And for this, there's several methods that are proposed. Now we see also in Lee presentation that we can apply machine learning and artificial intelligence. The main methods that are used in the literature that I published are focused mainly on trying to reduce the dimensionality of our data or applying these multivariate regression models or Bayesian approaches. Actually, in my team, we proposed 3-4 years ago, this extension of the multifactorial analysis, which is a dimensionality reduction method, in which instead of performing a principal component analysis for the decomposition of the data, we use independent component analysis. I don't know if you are familiar with this method, but mainly what we do is a multivariate PCA for trying to extract the principal component, explaining more variability of our data. Here, for instance, we can have 2 data sets, but also we can extend this to several matrices or data sets. And the idea is to normalize this data, combine this data, and when we have this combined data, to compute again a generalized PCA for extracting the components. In this method, we propose an extension, as I was saying, including not the PCA, the individual and the generalized, but also the independent component analysis. So we use the same, but with the independent components. And then at the end of the method, we include also a regression step, because in multifactorial analysis, at the end, when you extract the components, as in PCA, these are informative of the variability explained, but are not providing an inference in the analysis. So we propose this additional step, which is actually following the idea of the PCR, the principal component regression. And we perform this using, again, data from transcriptomics and genetic variants. Here, you can combine neuroimaging and genetics and use a clinical outcome, for instance, or you can combine different types of omics data and then use neuroimaging as your outcome of interest. And what we found actually was interesting combinations of genes and SNPs that were associated with these hippocampal measurements. Also in this work, we proposed a simulation study, and we were also seeing how we were increasing the variability detected compared with the multifactorial analysis, but also with the univariate analysis, taking those features that were including this linear combination of these components. So actually, we were increasing quite a lot the variability explained in these models. But actually, the last thing is that in all of these strategies, at the end, we are taking neuroimaging features as univariate components. We are not taking into account the joint modulation of the brain. And actually, this is something that we wanted to include in this type of studies, because at the end, our brain is going to, move and it's going to change relatively to the other, to all the regions. In this sense, what we were trying to propose is a strategy that is going to consider the brain as a component and each of the features as little relative variations or fractions of relative variations. So we are using in this study a compositional data analysis strategy. For instance, we use the alpha study data and the ADNI sample that maybe you are familiar. The alpha study is a study, a cohort study in Barcelona in our research center. And here what we did was to compute genetic predisposition of Alzheimer's disease and try to see, using this compositional data analysis, if there were an association of genetic predisposition and the joint modulation of these brain structures. The algorithm is a bit complex. I don't know if you are familiar with it, but the idea is to try to find and use a log ratio approach to find these different relative volumes that are, let's say, changing together. And this can be performed in a cross-sectional way or in a longitudinal way. We apply this first cross-sectionally, and we are extending the results longitudinally. And then, after defining the individual variants, individual regions that are changing together significantly, we try to assess, using a logistic regression, the association of if this is significant between individuals that are at high genetic predisposition of Alzheimer's disease or a low genetic predisposition. And what we found were interesting results. We classify individuals in the alpha study and ADNI following the Alzheimer's disease stages. In alpha, we only have cognitively unimpaired individuals, classify amyloid negative or amyloid positive. In ADNI, we took MCIs and ADs that were amyloid positive. And we found interesting results. We see here the different regions that were selected in this balance, in this relative variation. And actually, you can see that there's a little variation. So the amygdala, for instance, was more prevalent in MCIs and ADs, while the hippocampal volume or temporal regions were more prevalent in the cognitively unimpaired or preclinical phases of the disease. But also part of this, we really want to go a bit further, and we want to really extend all of this analysis as well, including other type of tests. So for instance, including non-parametric tests for when we analyze, for instance, white matter hyperintensity, so these types of features that are not Gaussianly distributed. We want to also use, as Lee was presenting, different types of classification methods, including not only MRI features, but also genetic components or PRSs, for instance. And then finally, we really want to go through the analysis of local ancestry, how genetic local ancestry is affecting all of these associations in relevant targets of Alzheimer's disease. And this is actually very important, and I wanted to show, for instance, this paper that was recently published, in which it was evaluated in different ethnicities, the association between genetic variants and hippocampal subfields, the subregions of hippocampal volume. And if you see here, depending on the type of subfield, we have different genetic variants that are associated to them, but also there were an important contribution and difference between the ethnicities that were included in this study. So with this, I want to thank my team, all the collaborators, and also all the alpha study participants, all the partners, and all the funding support. Thank you.